Great. Welcome, everybody. I'm going to test out. Great. We have 3 topics to go. Let's get started. So most of you have heard about some sort of particulate testing, either particulate matter, foreign particles, or visible particles. This is the common terms where you find these. When you analyze a product, normally you have different kinds of methods. One is the chromatographic testing portfolio for ID, potency, assay. And then there is contamination tests like residual solvent, elemental impurities, and all the particle techniques. And this is actually where the patient safety is. The contamination is the relevant part. This is what we are going to focus. And why are the particulates so much in focus? It's actually because sensitive parts of our body are in the same size range like the particulate matter that we are talking about. It's micrometer size range. Our blood capillaries are exactly in the size range. So whenever you get something, for example, injected, there is a potential health risk if there is a particle present. And then the other relevant point to consider in the patient risk is the administration route. Whether it's injected in through the muscle, for example, then it's more likely that the particle gets stuck in the muscle and doesn't move, it's located. If it's intravenous, it's more likely that it's distributed by the bloodstream somewhere in the body and ends up in whatever place with unclear outcome for the patients. And another motivation that's also always in the back of our mind is whenever there's a finding for particulate matter, it's also a quality and business issue. Normally, in the extreme case, when you find particulates, the worst case is you have to do a product recall. There is a list on the FDA website listing all the product recalls. There is quite a number because of some sort of particles or particulate matter that was found. And this is, of course, a very expensive matter. The regulatory background is divided actually in 3 parts. The first one is the drug product testing, which is quite nicely outlined in general chapters of the pharmacopoeia. Likewise, there are guidances for manufacturing suppliers like packaging material or single-use systems. This is equipment which may be used in contact with the drug product, and therefore also some sort of particulate matter limits apply to that. And these 2 elements are within the framework of control strategy and risk analysis to keep some overview on the particulate burden in your product. When do we find particulates? It's actually whenever testing is done. And it can be very early in formulation development, it can be during stability studies, it can be in release of batches. On the right side, you see an example. It's a fiber in a transparent glass vial. And normally, such a finding would cause some analytical investigation. And all these analytics are usually what goes into the analytical control strategy. This is an overview on all the potential sources where particulate matter might enter in your manufacturing. It can be in the starting materials, it can be somewhere in the excipients, it can come from manufacturing, it can come from some operator handling, and it can be from the packaging material as well. And all these different, oops, no, this wasn't too much, sources of contamination can be, of course, lead to a quite diverse particle population. You need some sort of classification. So this classification is done, for example, depending on potential patient exposure, or it can be the size of the particles, the particle type, the chemical compatibility or sterility, the amount per unit or container, and the frequency of occurrence, of course. Now we start with 3 different slides on classification. The first one is, obviously, you can classify particles by their size. The graph on the right side shows results from visual inspection. This is statistics that's publicly available. As you can see, for the size range between 50 and 500 micrometer, the results from human operators are quite much dependent on a probability of detection. For the smaller particles, the red is always what goes unseen, and the blue is what was detected. The smaller the particles, the lower the probability of detection. This is a thing we have to keep in mind. And this is where things come into account, like the morphology of the particle. Is it a transparent particle? Is it something that is maybe hard to spot, or is it something which is easily detectable? And the other thing we have to consider is sometimes the packaging is an issue when you do these investigations. If it's amber vials or even the dosage form, if it's something that's a somewhat opaque solution, can make it difficult to spot these particles. Good, thanks. Another way to classify particles is by where they come from, where the source is. Is it something that comes from the outside, like hair or fibers from human operators? Then it's extrinsic particles. Is it something that's coming from the manufacturing process, for example? Then it's intrinsic particles. And the last category, the inherent particles, it's usually something that comes from the drug product itself. It can be, for example, protein aggregates. And the third option for classification is by their chemical identity. In such a situation, for example, all protein particles go into one category, or all metal is one category, all the different steel alloys that you can have. And then there are many categories for all kinds of polymers that you may see. This slide shows an example of a particle library. As you can see, it's summarizing a lot of analytical results. If you use it as a part of your life cycle approach, starting collecting all the data on particles that you have ever found in this product, collecting these data and track and trend, then you have in the end a good overview and a good knowledge on the particle population that's potentially possible in your product. This makes it easier, or this history makes it easier to have quickly access in case of some investigation that's needed, to keep the overview and do the investigation quickly. This life cycle approach we have mentioned, I skip it. More interesting is the techniques for characterization as shown in this slide. When you get to know your new product, it's something you need to do is the characterization part. This is an overview on a potential workflow that can be used for quite smooth characterization with all the decision points that you enter there. And in the end, you get all the data that you need to understand your particle population, do a quick classification, and then later on you have the controls in place to spot these or that type of particles. With this, we come to the case studies. Our first case study is a parenteral protein therapeutic. What was observed was visible particles in solution. These particles were then isolated, and as you can see, it's something, well, it looks amorphous, it looks quite transparent. The IR spectrum that's shown there is showing the typical polyamide bands, the amide 1 and amide 2, which is quite characteristic. When you compare this observation in the microscope with the IR spectrum, the most likely characterization is it's probably some protein aggregate. And to prove that, and also to find out the source of this protein aggregate, we performed PEPMAP LC-MS investigation, meaning the particle was digested and then the amino acid sequence was compared to that of the drug substance that was used for this product. What we found then is that the particles were indeed drug substance, meaning this is an example of inherent particles, and adding this information to the control strategy is a relevant piece of information in the characterization work. Our second case study is coming from nasal and orally inhaled drug products. What is OENDPs? This type of products is usually coming in some devices. These devices are made of plastics, all kinds of plastics. And this is already the first hint. We have something in the device, we have to get it out. It's quite a challenge to get it out. Usually it's quite helpful and a practical approach to get it dissolved and do a filtration. And then a lot of different polymers can be found as shown in the different IR spectra and listed on the side. Because there is a contact between the drug product and the surrounding device, of course. This is our last example. This is a case of glass particles, more difficult to detect particle, definitely. Glass particles can come from many different manufacturing steps. It can come from the glass while it was used for packaging material. It can be from glass-glass interactions. It can be from many sources. And actually, glass particles are a quite common source of product recalls. So this is a relevant type of particles to characterize and to look at when you find it in your product. What we did was isolation by filtration. Then we did an IR spectrum. This is indicating some silica-containing material. It's inorganic, it's not that characteristic. And then we did with SEM-EDS the elemental composition. And this clearly shows a composition with elements that are typically present in the glass. And again, the combination of the visual appearance and the analytical data led to the conclusion this is a case of glass, even though it may have been hard to spot that. With this, we come to the conclusion. There's three main things we need to consider when talking about particles, particulate matter, and a reasonable control strategy to address this. It's the patient health and the risk for the patients. It's the quality and business impact, and then the analytical challenge. And with this, thanks for your attention and feel free to ask questions.